My title it got changed because one of our students that you heard from earlier this morning, Summer Z, actually her poster got selected for a talk. So I'm now talking about toward precision medicine and patient digital twins for NAFLD with a human biomimetic liver, MPS, and the BioSystics analytics platform. So this is the list of what I'm going to tell you this morning. First, structured liver MPS are important in the continuum of fit-for-purpose experimental models. Non-alcoholic fatty liver disease is a complex disease with no approved drugs. And the quantitative systems pharmacology, or QSP, and MPS approach has characterized NAFLD complexity, predicted and tested drugs to halt NAFLD progression. And then patient-derived liver iPSCs for NAFLD clinical trials on chips are in development. And 5, there's great potential for creating patient digital twins for precision medicine together with the BioSystics analytics platform. Why biomimetic structured human liver MPS? Why are they important? Well, first, liver is more than a metabolic engine with the hepatocytes. Liver is the primary or important secondary organ in multiple types of diseases. And the liver involves multiple cell types that communicate with one another in their microdomains, which in the liver are the different zones, as well as communicating with other organs. So we've set out over the last number of years to recapitulate the liver acinus as the functional unit. And it's characterized by having a vascular component, the acinus, which is lined with specific liver sinusoidal endothelial cells and contains resident macrophages, the Kupffer cells. And then the hepatic chamber contains the hepatocytes and stellate cells, as well as cholangiocytes and a few others. But we have focused on the first 4 cell types in the building of the initial model. The other important characteristic, as you do create microdomains with the flow of blood through the acinus, where the oxygen tension is distinct at the front end and much lower as the blood enters the central vein. So there's different physiology and pathophysiology that occurs in the zones. So biomimetic structured liver MPS are critical for mechanism-based knowledge. So where are we going with MPS and liver-based diseases? I think it's fair to say we all believe that in time we'll be using stem cells so we can make these precision medicine tools. These cells can be obtained either directly from the organ, and we make organoids that way, or you can access through the skin cells or blood cells and generate iPSCs that then can be driven to specific, in this case, liver cells. They can then be combined and allowed to self-assemble to make organoid MPS, or physically layered or bioprinted to make structured MPS, which is what I'm going to focus on today. Either one of these types of models within microfluidic devices can then be used for predicting disease progression and response to drugs. And very importantly, it's going to be a tool, many of us believe, for improving clinical trial designs before drugs ever see patients. You can study individual organs on chips by adding components to that chip that would arrive from other organs, or you can physically couple them to other organs, which we do in both a Nordis platform that we use the lamps on, as well as the vascularized lamps that we do with the microNIT platform. This is a summary of our human liver MPS-based programs, and the focus for today, which is not all of them. We are testing therapeutics for metastatic melanoma in the liver. We have an SBIR with DILIsym Solutions, which is a computational modeling company where we're looking at biologics-induced liver injury. We are finishing the paperwork to start a bioprinting grant for the V-lamps for NAFLD. We have a type 2 diabetes drug discovery program that also obtained some funding to look at COVID. But today I'm going to focus on the NAFLD drug discovery program, which easily couples to our program in optimizing clinical trials for NAFLD, and then coupling that to what is now the BioSystics analytics platform. NAFLD is a progressive disease, and it's a growing and global challenge, with 25% of the world population fitting somewhere in the continuum of NAFLD disease, characterized early on by an increase in fat droplets within hepatocytes or steatosis. A percentage of patients go on to exhibit non-alcoholic steatohepatitis, which is immune infiltration. Percentage of those patients go on to develop increased fibrosis that can end in cirrhosis, which is an end-stage disease. And some patients actually go on to hepatocellular carcinoma. One of the problems is this isn't a linear progression of disease. Different patients have different rates and can actually skip over different stages. So our initial goal in going after NAFLD using chips is to repurpose drugs and/or drug combinations that can halt or potentially reverse the disease phenotypes and genotypes. And we decided to start with a focus on stage 3 fibrosis and earlier in order to develop therapeutics. To add insult to injury, NAFLD has recently been proposed as one of the components of the metabolic syndrome that includes obesity, lipidemia, diabetes and insulin resistance, and hypertension. And many in the field have suggested renaming NAFLD to metabolic dysfunction-associated fatty liver disease. The prevalence of NAFLD is increasing worldwide. And in fact, if you look at the red, that's equal to 30% or more prevalence in the population. So there are two major problems. How do pharmaceutical companies optimize patient cohorts for NAFLD clinical trials? Because all of the trials so far have failed to meet endpoints. The second is, how will clinicians identify the right drug for the right patient? And obviously, this is a component of precision medicine and will be required to address the heterogeneity that's due to genetics, the environment, as well as the lifestyle of patients. Some patients are at increased risk of progression, for example, the PNPLA3 polymorphism, and that's something that Summer talked about earlier this morning. Patients have significant phenotypic differences due to the environment and lifestyle. And patient disease progression rates vary widely, and only a fraction of patients respond to specific treatments, hence the failures in clinical trials. Well, a key parallel approach to traditional target-centric high-throughput screening in animal models is something we've been working on for a number of years, and that's the use of quantitative systems pharmacology, or QSP, and human microphysiology systems as a powerful combination for drug discovery and development. As a reminder, QSP is determining the mechanisms of disease progression and mechanisms of action of drugs on multi-scale systems through iterative and integrated computational and experimental methods. So you can have an experimental platform, generate data, start building the computational model, make predictions from the growing model, test it back into the model, and do this iteratively. And this can increase and optimize the development of therapeutic strategies. Well, I don't have to tell this crowd about human MPS, but the key is that if you combine human MPS for any disease, we're particularly interested in the liver, with QSP, it is a powerful approach for drug discovery development clinical trials on chips, and the path to patient digital twins. So this is a simplified pipeline of what we do applying QSP and MPS. We always, we always start with patient material. In this case, in the study I'm describing, 182 patients that underwent bariatric surgery, and samples of their liver were taken for doing RNA-seq. That data can be used to infer pathways of disease progression. That data can then also be used to predict drugs and targets by machine learning and systems biology approaches. And then you can generate patient-derived primary cells and iPSCs and test the predicted drugs and candidates in human biomimetic liver MPS. And again, you can start building a computational model. In this iterative process, you can improve things before you even start doing specific drug testing. In addition to the experimental data, we also use validated published knowledge. This combination is then uploaded into the BioSystics analytics platform. And because QSP is a systems biology approach, you expect emergent properties, which can include novel targets, biomarkers, repurposed drugs and combinations, the starting point for novel therapeutics and therapeutic strategies. All of this knowledge then can be fed back to patient populations. So we have a human NAFLD liver MPS model that can be progressed from normal. And then we've predicted drugs and drug combinations to halt the disease progression. And we've started with primary cells as a reference, and now we're building the patient iPSC preclinical trial platform to make it personalized. These two models that we use, one is the LAMPS, or the liver acinus MPS, based on the Nordis chip, and the vascularized liver MPS based on the microNIT platform. There's pros and cons to both, and we use both of these platforms. The LAMPS model is very simple to make. It's very reproducible. You can control the oxygen, for example, and hold it at either a zone 1 or a zone 3 level. The VLAMPS is more complicated, more sophisticated. It has a vascular channel as well as a hepatic chamber. So in the vascular channel, which is lined with the liver sinusoidal endothelial cells, we can actually pass drugs and cells, including immune cells, that can then enter the hepatic chamber when activated. The first studies have been focused on primary cells, and as Summer mentioned this morning, we are developing the iPSCs for the others. A key element of this is we've built a panel of phenotypic and functional and genomic metrics to characterize the chips. I won't make you sit through me reading this list, but there's multiple components of the secretome. We have fluorescence-based protein biosensors in individual cells, and we have additional imaging metrics as well as endpoint measurements, including doing RNA-seq on the model itself. All of this data is uploaded into the BioSystics analytics platform, and I won't dwell on this because it's been talked about multiple times at the meeting, but we have found that it's critical because when you vary the cell line, the media, the matrix, any of these variables that we have in MPS models, and 6 months later, a year later, you want to go back and see how things changed, you can do all of that analysis within the database, as well as using it as a starting point for the computational modeling. So we've quantified the progression of NAFLD using these models. This particular data is based on the LAMPS model. Summer, for those of you who were here earlier, described the media where we can progress from the normal liver with kind of physiologically relevant glucose. We can drive it into early metabolic syndrome by the addition of more insulin and free fatty acids, and can drive it into NASH by a further addition of immune activation factors. This is a summary of some of the main metrics, and I'm not going to repeat all of the data that Summer showed, but we get steatosis increasing over the disease progression, oxidative stress, inflammatory activation. We get glucose, insulin resistant. Sorry, there was a little bit of COVID fog for a moment. Here are some specific measurements. These were all done in zone 3, what we've reported here, because these major characteristics of fibrosis, steatosis, and immune activation are actually maximal in zone 3 compared to zone 1. This work was led by Mark Meadle in the group. So this is kind of the QSP part of this. And we've done computational analysis of NAFLD patient RNA-seq data. And we've been able to define the molecular disease states and then use that to predict drugs for experimental testing within the liver MPS. This is obviously a complex image, and you can't see it well in the back. But what we did was to take the 182 patients, which are shown in this heat map in the columns, and then pathways within the KEGG space are the rows. And then we applied analytics to unbiasedly correlate all of these components. And then they sorted just using the data, again, in an unbiased way. And that produced from this pathway characterization 3 components: a quote normal steatotic group, a principally lobular inflammation group, and a principally fibrosis group. The metrics here are blue shows a lower than normal pathway expression. Red is above. And with this unbiased analysis, we can now use this data and build a pipeline of computational and further systems biology tools to get to the point of predicting drugs. And I won't take you through all of those details, but we use 2 approaches to prioritize the drugs that we've determined out of Drug Bank. And we now have 40 that are highly prioritized that we've started investigating. This just shows the proof of concept from doing this. This is just 2 of the drugs out of the top 40. And alone, they only partially change the steatosis, lobular inflammation, and fibrosis. In combination, pioglitazone in this case, with varinostat, a HDAC inhibitor, actually suppressed all 3, and under concentrations of the 3 drugs that have been used in other applications. So now we have started testing each one of the top 40 drugs and their combinations with the goal of finding the top combinations that we will take to the clinic, and also use the knowledge gained here, the knowledge gained here to do new drug development. Summer also talked about the genetic side of susceptibility, where PNPLA3 has a high effect on susceptibility for NAFLD. There are others, some that have a small effect. We decided to focus on PNPLA3. It codes for a protein known to have hydrolase activity towards triglycerides and retinol esters. Importantly, and this is a philosophy that I think is important, before doing iPSC-derived studies in CHIPs, we think that you need to define the adult reference point using primary cells. And so we've started by identifying in primary hepatocytes that were genotyped those quote low risk and the high risk PNPLA3 to do experiments on. So we've essentially set up chips that are in normal fasting media, early metabolic syndrome media, and then driven to the later stages of the disease. And in fact, just looking at the difference being the presence of the high risk component, we're able to see that intracellular lipid is increased when you compare the low risk in blue to the high risk in red, where you're looking at, in this case, total lipid tox that represents the amount of lipid. And you can also do the same thing with the secreted triglycerides. So this is a major component of the genetic impact. So we have a large NAFLD patient population that we follow in the UPMC Fatty Liver Obesity Wellness Clinic that supports the NAFLD clinical trial on patient CHIPS program. We start by taking blood samples from these patients where we have all of the clinical data on each one of them. Using their blood cells, we generate iPSCs, and then we take those and drive those to the individual cell types that we want, and they can be put into the chips to build that individual on a chip. So far, we've have 246 subjects with NAFLD NASH that have been enrolled in the program, and they've been genotyped. And this is just through April of this year. And again, we're identifying all of the variants, but experimentally, we're focusing on the PNPLA variants. We have the high risk variant and the low risk variant, and that's what we're focused on now with the early work on the iPSCs. Since we just talked about the BioSystics analytics platform, I won't dwell on it other than to say we can't do most of what we're doing and are planning on doing without a one platform that has a complete data pipeline from discovery to clinical trials all the way to building patient digital twins, from very simple things to more sophisticated things. So we think people, whether it's this platform or something else that will do something comparable, it's time that we start managing all the data so we can know what we've done from week to week, month to month, and year to year. An exciting new venture is creating patient digital twins. And we're doing that starting with the NAFLD Flow Clinic patients and the liver MPS that we're building. And with patient digital twins, you integrate clinical data from each individual patient. Obviously, the goal is to have a chip for each individual patient or for multiple organs that are coupled or whole body. It would be more sophisticated chips over time. That data taken together can computationally be used to create the patient digital twin for precision medicine. We think this is the future. If we think the mountain to climb to optimize iPSCs is a challenge, this is a massive challenge. But just like there were naysayers a decade ago about the potential value of MPS, I think I've heard some of that complaining since we've started talking about this. I would say wait 10 years, and I think we'll see that it's having an impact. This is just the same list that I started off with. This is what I told you. I have a disclosure to make since I'm involved in BioSystics. And this is the acknowledgements, great team within the Drug Discovery Institute, and we have our appointments in computational and systems biology, and then great collaborators across the university, both engineers, clinicians, and other kinds of investigators. This is our funding, and I have to advertise that we have positions available in the Institute for research assistant professor and a couple of postdocs. Thank you.